Good day, everyone. Today I'm going to give a small presentation on Introduction to Biostatistics. So these are the outline for today's presentation. So what is statistics? What is biostatistics? Statistics is a way of organizing, summarizing, and describing data and methods of drawing inferences and generalizing upon them. Once you get the results of after the inferences, you can get the conclusions of for your question. Then what is biostatistics? Biostatistics is the branch of statistics that is responsible for proper interpretation of scientific data generated in biology, public health, and other health sciences. Why need of biostatistics? Why do you need statistics in biology? First one, you need to have statistical literacy. You need to you need to be able to read and understanding and evaluate the scientific literature. For an example, if we consider this statement, the probability that a first-year bird will be found in the North Sea is significantly greater than for an old one. And also, you can see some notations here. If you don't know statistics, it's quite troublesome for you to understand what this statement is saying. This x squared is representing the chi-squared, and df is degree of freedom, and p represents the significance or the p-value. The second one is it is essential for scientific method of investigation. That means if you are going to address a research question, then you need to have some idea of developing the hypothesis and what kind of variables you will be needing and what kind of statistical inference you are going to use for that use for your research question. So how do we address a biological problem in a statistical way? First, you need to specify the research question that you need to be answered. So based on that question, you need to form a null hypothesis and alternative hypothesis. Then you need to design the study objective to test the hypothesis. That means you need to identify what is your outcome variable, what is your exposure variables, what are your confounder variables, mediator variables, and what types of variable are they, etc. Once you understand your data types and data variables, you need to collect the data in a way that the bias is minimal. How do we minimize the bias? We need to get the appropriate study design for that. To understand the study designs or get the study design, I suggest you to discuss that issue with a biostatistician and get help from the biostatistician. Then you can process and evaluate the data once you collect the data and get the apply the correct statistical techniques to data. Once you get the results, you can interpret and draw the appropriate conclusions based on the results. Before move on to any type of statistical inference or method, first you need to identify the types of statistical variables, type of data. There are majorly two types of data, descriptive and measurement. Descriptive or categorical variables have two types, nominal variables and ordinal variables. Nominal variables have no order between the variables. For example, gender, eye color, ethnicity groups, there is no order. But for the ordinal variable, there is a particular order among the values. For example, response to treatment, a like rate scale. A like rate scale will be like strongly disagree to strongly agree. So there's an order. Then the measurement interval or ratio variables. It has also two variable types, continuous and discrete. Continuous variables are height, weight, blood pressure. Discrete or count data is like we can count number of siblings, number of accidents per day. Then comes to types of statistical methods. We have descriptive statistics and inferential statistic methods. Descriptive statistics provide summary indices for a given data, and inferential statistics produce statistical inferences about population based on the information from a sample derived from the population. That means we estimate population values from the sample values. So before we move on to the other statistical advanced methods, we need to make sure we know the terms of statistics well. First one is population, because we always analyze data for a sample, but we are focusing on population. The problem is population is too large or too big. We cannot access all the data points or all the participants, patients in the population. Population is a group of individuals that we would like to know something about. But the problem is collecting data for the total population is very time consuming and it's so costly. So it's not feasible in practical. For example, if we consider uh, the population of women who has breast cancer in America, if you want to collect the data for that population, it will take immense time and effort and also money. So studying populations are always expensive and time consuming. If the sample is representative of the population, then by observing the sample, we can learn something about the population. So sample is a subset of a population which represents the population. For example, sample of women who has breast cancer in the state of Florida. So the sample, we consider the breast cancer women, the patients who has breast cancer in the state of Florida will represent the total population of women who has breast cancer in America. So sample is very important because it has the characteristic of population, qualities of population. So we need to get our sample size correct. The optimal sample size is often a compromise between what is statistically desirable and what is statistically feasible. It is always important get the correct sample size because otherwise our research uh, question will not be significant. So there are a few methods to calculate sample size. Majorly we use a pilot study or a study that had been used before this study to calculate the sample size. In general, the minimal sample size is around 30. So once we know our data and the statistical inference techniques, how do we going to calculate our values or the calculate those large mathematical equations? If you are going to do it by manually by using hand, it will take your whole life. So it's not feasible to do it by manually. Then we have some software programs like SPSS, Stata, SAS. But the problem with these software packages is they are quite pricey. Uh, so what can we do about that? We have open source software called R. R is a language for statistical computing and graphics, and it provides wide variety of statistical and graphical techniques. It's free, available on any platforms like Windows, Mac, Linux, Unix, and it's easy to customize and expand with libraries. Usually, when you download R, RStudio is a GUI which is very user friendly. Uh, you just have to have a little bit of logical thinking and coding experience. Uh, Many of the codes are in the internet, so you just can dig the internet and get through everything. So these are some useful links that you can download our studio and uh, and get some knowledge about that. So how to summarize measurement data? So there are like three methods you can summarize measurement data. Measurement data is continuous and discrete data. First one is central tendency. Central tendency is a single value that reflects the center of data distribution. So central tendency can be achieved using mean, median, or mode. Mean is the average of a variable. Median is the midpoint of that variable. Mode is the most occurring number of that variable. Dispersion or variation is the extent to which a distribution is stretched or squeezed. That means how dispersed, how far the data points around mean. Uh, Indices of dispersion can be range, standard deviation, variation, interquartile range. And the third one is distribution patterns. We need to check the distribution of the variables because in later stages, when we do the statistical inference, we need to make sure whether our data is normally distributed or not. So we can have like bell-shaped normal distribution or skewed distributions. Then how to summarize the categorical data? Most of the time, the categorical data are summarized using proportions or ratios. Then the next important step is statistical inference, hypothesis testing. So how do we do hypothesis testing? How do we create a hypothesis? For an example, if we consider this, does the pulse rate differ between the patients who were subjected to a stress test and patients who weren't subjected to a stress test? We call the patients who were subjected to a stress test as test or case, patients who weren't subjected to a stress test as control. So how do we derive a null hypothesis? Hypothesis is always about testing uh, a difference between two variables. Usually, we consider the central tendency variables like mean, median, mode to compare between two groups. So here we compare the mean of the test and the mean of the control to derive the hypothesis. So the null hypothesis is there's no difference in pulse rate between two groups. The two groups are test group and the control group. The alternative is there is a difference in a pulse rate between two groups. H naught is the notation for null hypothesis. Mu test is the mean for test group. Mu control is the mean for control group. And alternative hypothesis is denoted by H1. So after we derived our hypothesis, these are the inferential statistics tests for the continuous variable. We need to, once we understand our data is continuous, we need to make sure whether our data is normally distributed or not. If the data is normally distributed, then we can use these four tests according to the hypothesis. So the one sample t-test checks a mean of a group. Two sample t-test testing mean between two groups. Paired t-test is testing mean between two groups, but the observations are not independent. That means same patient appear in different before and after or two time points. One-way ANOVA is testing the mean among three or more than three groups. Then what if we have a continuous variable, but our data is not normal distributed? Then we have to use this non-parametric test. The first one, Wilcoxon sign rank test. It is similar to one sample t-test or paired t-test. Then Mann-Whitney U-test. It is similar to two sample t-test. The Kruskal-Wallis test is similar to one-way ANOVA test. How do we do inferential statistic test for categorical variable? We can use binomial test. It is similar to one sample t-test. Chi-square test. It is similar to two sample t-test and one-way ANOVA. Then McNamara test. It is similar to paired t-test. So this is a summary table of statistical tests that we can use in different hypotheses. I think this table is quite important. I hope you can go through this and understand the concepts of a little bit of biostatistics. I know this